Our next speaker is also a great friend of this conference. He's been with us 4 times already, because every time he comes, he brings us new news of his landmark discoveries in the quest for cures for cancers, heart disease, and osteoporosis. In write-ups of his work, I have seen references, and I do not exaggerate, to the fountain of youth and the cure for dying. He used to work here in TO at Amgen, but he was swept away by a titanic offer from the president of his native Austria, and now runs one of the world's great research institutions in Vienna. He is Josef Penninger. He's also a pretty good gymnast, and one of the times he was here, he, what do you call that thing? Somersaulted, cartwheeled across the stage. Yeah. Can you do another one?
We can do one together.
All right.
So you want me to do a cartwheel with Moses? Yeah. Maybe after my talk. So it's great to be back in Canada. I used to live here, and so I can really appreciate the culture, the humor of you guys. So it's really nice to be here again. So I actually wanted to bring you 2 entirely crazy ideas, which are not crazy anymore because we actually did it. And last year I got completely crazy because somebody gave me a phone call, Francesca Habsburg. So her granddaddy was actually the guy who was the last emperor of Austria, who actually started the First World War. And Francesca said if I want to come with her on an expedition to Galapagos. So I actually learned diving, which I've never done before because of fear of water. And so this was my 8th dive in my life, a whale shark. So I was completely nuts. And so this is me down there. And trust me, it's, sharks are spectacular. But actually, if there are 400 sharks around you, for somebody who grew up in the mountains, it's not that spectacular anymore. And in particular, if you get a nosebleed. One day I actually had a nosebleed, and so I was this close to being eaten by sharks. I'm still here, and so I can tell you my little story. And the pictures I show you actually are from, great, thank you. So the pictures I show you are from a fellow traveler from the Dominican. So these are not mine. So what I'm actually doing, I'm a genetic engineer. And being on Galapagos, one sees evolution. A reverse evolution, birds which stopped flying because they had no enemies, progressive evolution. And basically, I do genetic engineering. Since 20 years now, we manipulate genes in stem cells, create new organisms, and basically ask the question, what's the function of the gene? So can we read and make sense out of the genetic universe? And in my first IdeaCity, when I was invited here, we'd actually just created a new mutant animal in a gene we call ranked ligand, and we actually at that time hit the jackpot, because what we had found was the master gene for bone loss on this planet. So to have a healthy bone, there are 2 cells which make it, one which adds new bone, one which eats bone away, and 95% of all bone diseases, and if it happens on the whole body, we call it osteoporosis, are because these bone-eating cells eat too much bone. If it's local, people with rheumatoid arthritis or cancer, patients with metastasis into the bones, it leads to bone loss. And in most of the cases, it's the osteoclast, the cell type, which starts eating bones. And with this little genetically modified animal, we basically found the master regulator and the controlling element of essentially all bone loss regulated by bone-eating cells on this planet. So this was quite interesting. And I actually proposed, based on this fundamental principle, how our bones work, for all of us as is fit here, for all other animals on this planet, one could actually design and engineer the first rational therapy against bone loss. And now some years later, you can actually go to the doctor, you can get the shot of this molecule. It's a monoclonal antibody. It's probably the most potent molecule on this planet to shut down bone loss. And it's approved now for osteoporosis. Just to remind you, osteoporosis is one of the most frequent causes of death on this planet. Old people, in particular in aging societies. If we actually would like to go to Mars for holidays, it might be nice, but none of us would be able to walk anymore. Now we can, because you get the shot of this little medicine, it works. The medicine has also been approved already for more than 1 million people a year who get metastasis to the bones, women with breast cancer, males with prostate tumors, you name it. In many cases, the cancer leads to bone breaks, vertebral breaks, people get paralyzed if one shuts down, of course, the molecular principle. How bone loss happens, one can treat this rationally. And just recently, there was a study published, which was really cool, a horrible tumor called giant cell tumor, which is and has been completely untreatable. It leads to deformation. In many cases, you actually have to cut off the arms or the legs of these poor kids and or adults who have it. Turns out this tumor is actually hooked onto this molecular principle. If you give them the drug, basically everybody reacts. So we made an untreatable cancer to something which is perfectly livable for. So when we did this, we also realized the one reason why women get more bone loss is because the same molecule actually doubles in function. And it's the molecular principle how particular female sex hormones talk to the mammary gland and in pregnancy tell the mammary tissue to proliferate. And so we had this idea, this could be actually a missing link, how sex hormones in the pill and the hormonal replacement therapy drive breast cancer. So it took me 10 years to do the genetic engineering to actually prove the idea. We have it now, and indeed we published this recently. This is the way we believe, and actually others believe now, how sex hormones drive early breast tumors. And of course, knowing the system, having a medicine which is approved, one can actually now run, and it's already being done, huge clinical trials for prevention of potential future breast cancer. So this little mouse we genetically engineered when I was still in Canada here basically gave us completely new understanding how bone loss happens, how breast cancer develops, why sex hormones talk to the bone and to the mammary tissue. And of course, knowing the principles, we can design and have been designing new medicines against it. So when we did this, so I actually got an innovator award from the US government, so it was quite interesting. They're sending $7 million from the Department of Defense. I think it's very good money of military money to be spent to Vienna to do breast cancer research. And so we started to think a little bit on fundamental principles or new ideas, how one could treat cancer. And we were thinking about molecular evolution and evolution in the immune system. So we have these hundreds of millions of immune cells, which are basically scanning all the time the environment, if we have a virus infection, if a bacteria is in us, to fight it. However, over hundreds of millions of years, this immune system has also been sculpted to see something which is not normal in our body, something called immunosurveillance. So the idea is every second as we sit here, our cells, which are trillions of them, some of them become cancer. But our immune system can see it and kill it. And of course, if we would understand this balance, which allows them to kill the tumors, then we could interfere with it and make basically completely new medicines for cancer, which not work for a few people, but basically for essentially every tumor in the future. So we came up with this idea. We actually found one, and it's not important. Other people finding other molecular breaks and checkpoint control molecules. And we call this little one CBL-B. This is actually an experiment of metastasis. Turns out when we release this break, we can reduce metastasis massively. You can just look at normal metastasis in here. There's actually all animal experiments. And then we thought, knowing the molecular principle, we could actually make a small molecule, a new drug, with the grand vision that one day we have a pill you can just eat. You listen to me at IdeaCity, you pop your pill, and it actually reawakens your immune system to kill the tumor. With relatively few side effects. So we did it. We just published it in Nature, and it really works. So this is a mouse experiment. We gave the mouse metastasis, gave the mouse the pill against this molecular checkpoint of the immune system, and we can indeed license immunity to kill cancer. So, and of course, other people who work on this, and it became really a new paradigm of cancer treatment, because now what people see, big companies are all getting into this, is long-term survival of some patients. So in cancer, we probably will never find the magic bullet to treat it, but to make it a chronic disease with less side effects. And in some people with these new treatments, they really have long survival advantages. So let's see if it works. This is not just theoretic. Next month, the first humans will actually get our new idea. And then we had some totally crazy idea, which I want to tell you. And this idea was, it took us years to come up with mutant animals, find molecular principle, and so we thought, wouldn't it be beautiful to mine the entire genome in a day? Check out every single gene that's function in a day, whatever we can model. And this was, of course, completely gaga, because how you would do this. But there is an organism which does this, and this is yeast. So if you drink your beer, this is what ferments your beer. And at a certain stage of its life cycle, they have a single set of chromosomes. And for us, as a population, to actually stay healthy is because we have two sets of chromosomes, one from our mother and one from our daddy. And when the sperm and the egg get together, we have two of them, and basically every single cell we have is diploid, has two sets of chromosomes. But for us as geneticists, it's a real problem, because, of course, if we make a mutation here, the other one is normal. This is why biology and nature actually wants outbred populations. So we actually stay healthy, so they want me to find a Chinese girl, so to spread our genes, so because it's very unlikely we carry the same mutations. So the dream was, could we make a mammalian cell with a single set of chromosomes? I thought this was crazy. This was against the paradigm of biology. This cannot work. People tried it. So my student came to me, proposed it. I looked at him and said, this is the most crazy idea I've ever heard. Go back to the lab and do it. So, so he went back to the lab, and this was the idea. Something we called pathogenesis, immaculate conception. Because in our body, of course, two cells, which a single set of chromosomes, the egg and the sperm. And so the idea was, if we could trick the egg to believe there was a sperm, also there was no sperm, they would make a little embryo, but they, of course, come from a cell with a single set of chromosomes, because we did immaculate conception. And it's very easy, actually. You just take 5% ethanol for 10 minutes. It really works. I can explain it to you why it works, the molecular mechanism in details, but you probably don't want to hear it and don't care. You can also use strontium chloride. So we basically took an egg with a single set of chromosomes, tricked it to believe there was a sperm, they formed a little embryo, and out of them we grew stem cells. And this is the first stem cell on the planet which has a single cell of chromosomes. We grow them now since 3 years in the lab. This is the guy who did it. As you can tell, he himself prefers traditional cloning. So these cells are stable. We can make billions out of them. They're stem cells, so these are stem cell markers as we know it. And we can even make mice out of them. So these are basically mice which have never seen a daddy. So And 2 Chinese groups after us, so this whole field is now exploding. 2 Chinese groups after us actually showed you can actually make mutation and pass it on the next generation. So only coming from a mom. So immaculate conception, so we are not Jesus, we just do it in the lab. And this is the reason why we do it, and I explain this to you. So we can do now play forward genetics, the grand vision. Can we mine the entire genome in a day? Because now we have a cell with a single set of chromosomes, and it's a normal cell, it's a stem cell, we can basically make anything out of it. And of course, if we mutate it, then we could mutate basically the entire genome in an hour. So we spent the last 4 years to develop systems like this, jumping genes. One of them is called Sleeping Beauty, so we basically put Sleeping Beauty in the cells, and an hour later, you have 50 million mutations. So we can play whole genome genetics in a day. So my students are now doing basically whole genome scans in 4 weeks. What has been took decades for hundreds of people to do because of this little new technology. And the idea was, we grow them, we mutate them, we select for something, we go to sequencing machines, and then you know exactly what's working. We use viruses. We were clever about it. You can repair the mutations. I can explain to you how it works. So we have repairable whole genome mutations. We barcode them. It's like going to the supermarket. You scan the barcode, you know the product, you know the gene, and you go. So one example, ricin. It's one of the worst bioweapons on the planet. Has been actually used in the First World War to spread it over the soldiers of the enemies. Last year, Obama got the letter of ricin, the mayor of New York. A single molecule kills a cell. So if you inhale it, you're dead in 2 days. There's nothing which helps against it. And we thought, this is a great technology, let's use it. I got some really interesting phone calls afterwards. And sometimes it's actually great to work in a little country like Austria, because there was no way we could have done this in the US, for instance, probably also not here. So that's how it works. We take our cells, single set of genome, we give them this poison, they all die, you see they're all dead here. We take the cells, give them Sleeping Beauty to make 50 million mutations, or a virus, or AIDS-based virus, whatever we can engineer. Make mutations, cells grow out. The assumption is now we hit something which is essential that the poison kills, because otherwise all cells would be dead at the whole genome level. 3 weeks later, we go to the sequencing machine and we have the whole pathway. So this is something else we have been doing now. Artemisinin, the best medicine against malaria, 100 million prescriptions a year, is very toxic. Nobody really likes to mention this, has severe side effects, and nobody knows how it works. So we drop it on, 3 weeks later we knew how it's functioning. So we work now with some cool ideas on snake poisons and spider toxins, which kill everything. Nobody understands how it works, and so we are doing this. Whole genome genetics in a day, based on these little tricks of immaculate conceptions in stem cells. And we can play reverse genetics. Thousands of scientists have found cancer genes, genes which might be important in brain, in Alzheimer's disease, how nerve cells die for Lou Gehrig's disease, you name it, or how blood vessels develop. And so we decided, let's do something about it, go from known candidate genes to figure out which ones are real. So what we have been doing now in Vienna is, so we want to create for everybody in the world a Wikipedia of ES cell clones, embryonic stem cell clones, which are mutated in a repairable fashion with a barcode, where every single gene is shut down. So basically the entire genome, as we know it, this is mouse now. Of course, one day we want to do this in human. We are not there yet. We have shut down the entire genome. It's sitting in our fridges, and then you can go there and say, these are my 100 candidate genes I believe could be important how blood vessels develop in a cancer, because cancers, of course, need to have new blood vessels to get nurtured. So you go to the fridge, you sow the cells, you make new blood vessels with the mutations, so you repair it and see how this works. We are advancing, so we already made 70,000 of them. So we have now robotic streets, so we make 10,000 ES cell clones a week. Just a fast example. So we can grow blood vessels out of the stem cells, so this is how it looks. We can color them, so they look a little prettier, so these are blood vessels growing out, and we can mutate them and now see how it works. So it's possible to play evolution in a day in a stem cell. Of course, stem cells have the potential to become basically everything else. So they're all sitting in our fridges now. So this, I predict, and already people are using it, should completely revolutionize how we do functional genetic studies in the future. Lots of people are getting in this field. My postdoc was the first who did it. It goes very fast. It's super cheap. We can hit the whole genome in a day. We can grow billions of cells. It costs literally the 3 little plates in our tissue culture hoods. So where will this take us? Just a little example. I work for Madeleine Lancaster and Jürgen Knoblich. He's my deputy director in Vienna. They started to think about creating bioreactors. Because at the end of the day, we would like to combine functional genetics with tissue engineering. We grow our own tissues at home. So we don't have to take it from dead people, but we take a stem cell and grow a new tissue. And what Jürgen did, and Madeleine, she actually just won last week a huge prize, the best prize in Nature for her work. So bioreactors, it takes them, you go from a human, this is now human, stem cell, and you see these little things here, these little white organoids, and when you section through them, it's a brain. So what Jürgen did, he opened Pandora's box of brain research. He grew the first human brain in tissue culture. So I was involved. We actually made stem cells from children which had mutations, so we can model disease. And the reason why we do this, it's for the first time where we can actually, or might be able to model Alzheimer's, Parkinson's disease, brain cancer, schizophrenia, autism in a growing brain in our laboratories. So they live for half a year and around half a centimeter big. So that's our grand vision, to trash the entire genome in a day and combine it with engineering of completely new tissues. And this is, of course, interesting because the brain is the most complex tissue on the planet. And what's really interesting is, at a certain stage, the cells know how to find each other. They have this self-fulfilling way to get self-organized. And we're just talking now to IBM because they want to learn how brains develop for the new circuits, how they actually use new computers, how they program new computers for new data storage, and so on. And these brains are not just brains. You see these little things here. They actually fire, they talk to each other, these cells. So this is basically now going on in your brain. These little brain cells and these brain cultures are working. So, and now end here, I overstayed my welcome, I can tell. So I hope I did not scare you too much. But this is what we have been doing. If people in 500 years look back to our times, I am convinced they will say this was the renaissance of biology, the renaissance of understanding how stem cells work and how we can use them. Amazing new technologies. 5 years ago, we could not even have dreamed about to grow a human brain in tissue culture or model evolution in an hour using our new technologies. And I will use it, and I will give this technology to everybody, so this will be free for everybody to use in the future. And last week, I was at the funeral of a friend of mine who died of breast cancer. And that's the reason why we do it. So one day, using these technologies, we hope to come up with new medicines to help somebody. So thanks for listening.
An amazing guy. I can't let you go with asking a few questions, so I want to go back to practical matters that may involve everybody in this room. 300 million women worldwide suffer from osteoporosis, and arthritis, I think, touches practically everybody. Certainly, what, 1 in every 2 people, 1 in every 3 people. So when you discovered the master control gene for osteoporosis, does that extend as a possible remedy for arthritis as well?
We published this. Yeah. We have these mouse models and rat models of arthritis and crippling. So if you block the system, you can basically wipe out crippling in arthritis. Yeah. Because it's the same principle, always works. You know, it's like a funnel. You have 100 reasons how you turn on bone loss in the whole body with hormones, whatever, because you don't get off your butt, you go to space, or there's local inflammation where people lose their teeth, but all of them have to go through this funnel. And of course, knowing the funnel, we can block it and have rational therapy for all of it.
So is this medicine we can buy now?
Absolutely, yeah.
Oh. And it's called Rankle?
No, it's called denosumab. It's approved in North America and Europe for osteoporosis and cancer, skeletal-related events in cancer.
And can be used for arthritis as well?
There's no phase 3 clinical studies, so it's not approved for arthritis yet.
Really? That would be a monster bonanza drug. I have this complaint. You grow up thinking the hardest parts of your body, therefore the parts that are likeliest to last the longest, are the bones, right? Not the case. These lips, they're in good shape. They're working. The earlobes, huh? Full of life and pliant and buoyant and just waiting for somebody. But the knees, I mean, especially if you played a little squash, they're like a minor disaster.
Yeah.
So hurry up.
The bone loss is done, so we move to the next phase.
It's done. I bet we get everybody here to sign up for a little of that medicine. I wanted to tell you, Josef has a million accolades to his credit, but this is another just in. Josef has just been given the Wittgenstein Award, which is in recognition and support of excellent scientists who have already produced exceptional scientific work and who occupy a prominent place in the international scientific community. 1.5 million euro. Josef.
Thank you.
And I have one more question that I'm sure everybody here wants to know. Have you ever met Arnold?
I have not. But once, if you give me the time, when I was in Canada, when I was a young group leader, I was eating in the cafeteria, and this pretty nurse from the next table came to me and said, "Oh, I really love your accent. You must be Russian." So I looked at her strange and said, "Why do you think I'm Russian?" And she says, "Because you talk like Arnold Schwarzenegger."
That's great. Thank you so much.